But we're very honored to have Dr. Sintim here. He's the drug discovery professor of chemistry at Purdue. He moved to Purdue from University of Maryland back in 2006. Did you move there? 2016. Okay, sorry. And then so he was at Maryland for a while, and then he moved there. But he's also now rotating through the National Science Foundation, and so he and I share having Ponch as a boss. So I had him first. Now you have him. He's won numerous awards here, including the NSF Career Award, the Camille Dreyfus Teacher Scholar Award, the Sigma Psi Distinguished Lecturer Award, the NOVCCHE Lectureship at UPenn, and Purdue's Most Distinguished Faculty Member in 2019. And the list goes on. He's also started a company. For our purposes, we have been for a long time interested in this. As you know, biodesign is very interested in world challenges, and this problem of superbug resistance, these bacteria that can tolerate just about every antibiotic threat them, is a huge issue for the planet. Not to mention resistant cancers, which is an interest of mine. I know these are all interests of yours, so we're very interested to hear what you're up to, and I'm going to hand it over to you.
Thanks for the introduction. And
if you feel comfortable, you can take it off the mask if you want to, because you're at the front of the room. We'll all be behaved.
We're on social distance.
Okay, so thanks for the invitation and also the reception that I have received so far. This is a really beautiful campus, and the buildings, like different buildings with different architecture. It's really nice here. So for today, I'll be talking about a project. It's one of the projects in the lab, one of many, but it's a project that is very dear to me because in 2005, when I was looking for a faculty job, I sent an application to many places, interviewed at a few places, and I struggled to convince hiring committees that cyclic dinucleotides are going to be the next big thing. So I hope that by the time that I finish this talk, I would have provided examples to show that they are not going to be the next big thing, they are already the next big thing. Our interest started in bacterial, and we were interested in coming up with different tactics to kill drug-resistant bacteria. But it turns out that when you start working in bacteria, inevitably one gets into inflammation. And so now we are also looking at inflammation and also cancer. So I'm going to talk through a journey and show some of the contributions that we made to this really exciting field of cyclic dinucleotide signaling. I will make this proclamation that nucleotide signaling is one of the most important fields in biology and in medicine. And so we owe so much to L. Sutherland, who was awarded a Nobel Prize in physiology and medicine in 1971. But he was the first to show that a very interesting looking molecule called cyclic AMP, cyclic because you can see here that the phosphate is cyclic, and the nucleobase is adenine. So he showed that hormones, which we all know play important roles in biology, work through this simple looking molecule, okay? So he showed that signals from one cell to the other are conveyed by a messenger, the messenger being the hormone, and how the signals within the cells are then conveyed to another messenger. So cells have to talk to each other, right? And when cells stop talking to each other, bad things happen, right? So for example, cancer cells are deprogrammed, and so sometimes they don't listen to their neighbors. but this is a cyclic mononucleotide, and that led to a Nobel Prize. A few years later, another cyclic mononucleotide also led to a Nobel Prize, and Ferry Murad actually is a student of El Sutherland. And so they got the Nobel Prize for discovery relating to nitric oxide signaling in the cardiovascular system. But what really it was that this second cyclic molecule, cGMP, was at the heart of this mechanism. Okay, so again, is cyclic because you can see the cyclic structure here is guanine and it is a monophosphate. Okay, so when nitric oxide signal is produced, it activates the guanylate cyclase. This is the enzyme that takes GTP and basically cyclize to make cGMP. So 2 mononucleotides, 2 Nobel Prizes. Now, over the next few decades, several scientists then filled in the gap, right? So these mononucleotides were discovered, but the question was, what are their receptors? Once they bind to the receptors, what do they do? Are there opportunities to actually hijack these systems and produce novel therapeutics? And so now we can sort of have this schema here and show how a simple second messenger such as cyclic AMP, right, upon a stimulatory signal leads to this enzyme, adenyl cyclase, making cyclic AMP. And this is a second messenger, and it does so many different things. It binds to so many different proteins and enzymes, and it regulates so many processes in the cell. But in biology, most signals will have to be terminated, right? And so the enzyme that terminates the action of cyclic AMP is a phosphodiesterase, so we call this PDE, that will terminate and now make it the non-cyclic form. So this is just showing the works of many researchers. Cyclic GMP, which is also an important second messenger that led to the second Nobel Prize, also binds to so many different partners. And again, you can see here that the action of cyclic GMP is terminated by a phosphodiesterase. Cyclic GMP controls smooth muscle relaxation. And so it turns out that inhibitors of PDEs were developed for the treatment of various cardiovascular conditions. And the clinical trial was really interesting. So I'm not entirely sure this is what happened, but this is what I've heard. That many people were giving the drugs and it wasn't working for the cardiovascular indication. But when they asked for the drugs back, they were not willing in giving the drugs back. So the doctors probably thought that something was going on. But to cut a long story short, it led to trying this molecule for erectile dysfunction. And I will show the data for how much money is now made in just the inhibition of PDEs. But before that, I want to go back to these Nobel Prizes, right? So Sutherland, 1971, discovery of cyclic AMP, Ferry, discovery of cyclic AMP. But then when I show the scheme, there are so many processes, right? Just discovering the molecules are not good enough. You have to understand what the molecules are doing, what are the partners, etc. And so about 7 different times, right? 1, 2, 3, 4, 5, 6, 7 Nobel Prizes were given for work related to cyclic mononucleotide signaling. And we also consider that each award actually awarded multiple people. I would argue that there are not that many fields that you will find that many people get a Nobel Prize, right? So that is telling us something. And let's look at just one target in the signaling, which is one PDE. And so PDE5 is an important target because it degrades cyclic GMP. And when the PDE5 drug was first approved by the FDA, you can see here that Pfizer was really making, right? Almost $2 billion. Now, over the years, the sales have gone down. It doesn't mean that people are not using it. It just means that generic versions are now free, okay? So just one target. Now, there are many PDEs that our companies are working on for cardiovascular, ocular, etc. And so cyclic mononucleotide has basically led to billions of sales when it comes to drugs. So this is really interesting, right? Very, very important signaling molecule. So they always say that history is actually a good predictor of the future, which is quite odd, right? Because you always want history to be behind, but it turns out that history keeps repeating itself. But sometimes we don't learn from history. And by the time that we learn from history maybe we should have. So what really surprised me when I was looking for a faculty job was that I was looking for a job in 2005. And it turned out that almost 2 decades ago, there was a seminal paper published in Nature. And that paper disclosed for the first time this odd-looking molecule, which is called cyclic digenine, right? So it's cyclic because you can see here that the phosphates, right? You have a cyclic structure. This time you have 2 phosphates, so it's a di and it's guanine, so 2 guanine. So this is the reason why we call it cyclic digenine. It was published by Benziman from Israel. And what he showed was that in a bacterium that makes cellulose at that time, it didn't show that to be relevant to medicine, but to agriculture. That there was an enzyme called diguanylate cyclase that took 2 molecules of GTP, condensed the 2 molecules together to make cyclic diGMP. And this cyclic diGMP acted as a second messenger. But what it did was that it basically bound to an allosteric site of this enzyme, cellulose synthase. And so it activated the cellulose synthase to make more polymer, right? But just as we saw in the cyclic mononucleotide area, there is a phosphodiesterase called PDEA that breaks this molecule because it's cyclic di. So the PDEA will first break it here, right? And so you have what we call P, which is a phosphate, and then G, PG. So that is the linear, but it's still got 1 phosphate linkage. So Benziman identified PDEA, but in their paper, they indicated that there must be another enzyme called PDEB that will degrade this linear PGPG to the mono GMP, and that will terminate the action of the second messenger. So the identity of PDEB was unknown at that time. But it wasn't this detail that really bothered me, although we did look into that. But what bothered me was that cyclic GMP, right, led to a lot of really exciting discoveries in medicine, billions of sales in drugs. And this discovery was almost hidden. Now, there were some few microbiologists who were working on this, but from 1987 to 2004, before Benziman died, his lab and a handful of collaborators who worked with him were the only labs publishing on cyclic dinucleotides. So basically, the world just missed this. And then in 2005, Gal Perrin, who's a bioinformatics person, did a survey. And he published a very seminal bioinformatics paper. And in his paper, he indicated that when he analyzed the genome of most gram-negative bacteria, they all seem to have an enzyme that appeared to be a diguanylate cyclase. So the question is that why would this be so prevalent in so many bacteria if it wasn't important? And so that almost sort of spared the field, and many microbiologists jumped onto this And me being a chemist, I saw an opportunity. So I'm going to talk to you about these opportunities and how we provided some real contributions to the field. But I would like to return to this identity of PED. And so when I started my career at the University of Maryland, a good friend of mine, Vincent Lee, had also started. He had finished his postdoc at Harvard, and he was in the microbiology department. And so my mentor, Phil Bishon, basically said to us that, oh, Herman is interested in bacteria. You worked on bacteria and some of the cyclic nucleotide. Why don't you work together? And so we worked together, and that led to this paper that was published in 2015, where we showed that an enzyme that had been labeled as oligoribonuclease in many gram-negative bacteria is actually the long sought after PDB that degrades PGPG. So this paper was actually recognized as one of the breakthrough signaling papers in 2015. Okay? So what it means is that the whole pathway in this bacteria is at least sorted. Okay, so what do we know now? So 1957, the mononucleotide, and then look at that, 10 years later, the dinucleotide, and then the field went silent until 2012, another dinucleotide was discovered. So see that this is guanine and guanine, but here you have adenine and guanine. Okay. And then a year later, there was a seminal discovery. So this is common in mammalian cells. Cyclic GMP was found in bacteria. Cyclic GMP, 3' found in bacteria. I would like to return to this nomenclature, what is 3' means, okay? So if you look at the sugar linkages, you have 1', 2', 3'. So you have 3', 4', 5'. So this linkage is called 3', 5' linkage. So if you look at this molecule, it's a 3', 5' linkage, and this is a 5' and 3' linkage. So that's why we call it a 3', 3'. So bacteria makes a 3', 3'. Okay? But in 2013, it was discovered by Zichan Chen that mammalian cells also make cyclic dinucleotide, but one of the linkages is actually a 2', 5'. So this is the reason why we call this a 2', 3' cyclic GMP. Okay. So it appears that this molecule is actually linked across kinases. So let's now go through this journey, what our contributions have been. So our first major contribution was that we realized that if you cannot detect something, it's very difficult to study, right? Okay. So we thought, what I cannot detect, I cannot study. The field was using very tedious, low throughput, expensive LCMS assay to study cyclic dinucleotides, and some were even using radiolabeling. And so, for example, this was an assay that we did in a group that took a lot of training to do. We were using radiolabeled P phosphorus to label our cyclic AMP. So when we wanted to study this PDE, we could just do a radiolabeled TLC. Okay, very, very tedious. And so we saw a gap. So the gap in technology actually led to my first grant as a faculty, which was my NSF career award, where 3 years after I was able to secure a faculty job, I was able to then convince reviewers that this is going to be important. We don't know how to detect them. So one has to come up with a very simple way of detecting these molecules. And so the gap in technology was just a simple and specific detection of these cyclic dinucleotides. But we made an argument that once we could do that, then we would be able to use that in high throughput manner to find small molecules that inhibit these enzymes. We had our eye towards maybe in future, maybe also discovering Viagra type molecules, right? So what did we do? Cyclic dinucleotides are also nucleic acids, right? And we know how to detect nucleic acids. So nucleic acids can form very intricate structures. So for example, if your nucleic acid has a lot of guanines, then they can form something called G quadruplex. So I want you to take note of this, right? So 4 of them would come together to form G quadruplex. It also turns out that you can even have 2 cytosines also come together. You can have 2 adenines also come together, right? So there are all these non-traditional wax and crate-based pairings that have been seen in nucleic acid. But what you should note is that short oligonucleotides do not form stable higher order structures. Okay? So if short oligonucleotides do not form stable higher order structures, then how can we utilize the knowledge that we also have about nucleic acid and apply them to just a dinucleotide? So one of the early contributions that we made was that we recognized that the dinucleotide is really special. The cyclization endows these molecules to adopt certain structures that we could actually take advantage of. And so in this JACS paper that we published in 2011, we demonstrated that if you take a cyclic DMP and this phosphodiester, right, X is oxygen, which is cyclic DMP. We argue that if you change this image to sulfur, right, you would just have changed the bond lengths by just a little bit, okay? But this really small change, we argue that it was enough to change the conformation. So cyclic DMP can exist in the open conformation or the closed conformation, right? So we argue, just by doing computational work, that cyclic DMP existed more in the closed conformation when compared to the sulfur analog, right? And the sulfur analog. So here we've just done a very small change, and we are showing that this small change is affecting those conformations. So the supramolecular chemists amongst us will realize that if the molecule can form this, then it's almost like a cleft, right? And so that then opens up many interesting possibilities to be able to detect this molecule. Okay? And we then came up with this scheme that if cyclic DMP, because it's cyclic, if it can form this conformation, then you can have a situation where 2 cyclic DMPs could come together to form this intercalated dimer. But because it is prearranged where the nucleobases are in the same direction, it means that it will be very easy for this to form a tetramer, right? So you can see the G quadruplex here. A dinucleotide doesn't form a G quadruplex, but because it's a cyclic and it's got the right number of atoms, it can form this tetramer and can even form a higher order octamer. If this is true, if this scheme actually is valid, then there are many opportunities, okay? So the prediction is that cyclic DMP will have a higher propensity to form the tetramer, the T, and octamer structures than the endo-S, which computationally we have predicted. And if that is true, then we should be able to put an appropriate fluorescent dye in between these. So it's almost like we have a cavity, right? This molecule has a propensity to form these structures. So we could put in a fluorescent dye, and if we can do that, then we have a scheme for simple detection, okay? But if this is also true, if it is true that we can use a small molecule to stabilize a tetramer, then perhaps we can also use a small molecule to quench the signaling of cyclic DMP to small molecules, because it is the monomer and sometimes the dimer that actually binds proteins. So what this means is that in this scheme, there are 2 possibilities. We could come out with a simple detection of this molecule, but we could also go in and disrupt cyclic DMP signaling. Okay, so we made this prediction in 2011. And we did a lot of NMR studies to really show that indeed cyclic DMP forms these aggregates. I'm not going to go into the NMR experiment in details, But in NMR, there's an experiment, you can do something called a dose experiment, right? You can look at how a molecule is actually diffusing, and you can use that you can calculate the diffusion constant and be able to predict the diameter of the molecule. But what I want you to pay attention to is that this is cyclic GMP, and this is the endo-S. The only difference is that we basically change this atom here from oxygen to sulfur, a very conservative modification. And look at how it affects the aggregation state. In cyclic GMP, you see the octamer is the O, N is the monomer. So you see that the monomer doesn't even exist in such a high concentration. All you see is the octamers and the tetramers. Whereas you go to the endo-S, you don't see the octamers, you don't see the tetramers, right? So this signaling molecule is uniquely structured so that it forms aggregate. But because we saw the tetramer, we knew that we could put something in there. So we reported the first simple fluorescent detection of cyclic GMP in this JACS paper, where we show that a fluorescent molecule such as 5-orange, this molecule is typically non-fluorescent in solution because you have this fast rotation around this bond, so you have deactivation. But the idea is that if you can put it in a cavity, then you stop the rotation. And we had already established that this simple dinucleotide can form aggregates. And so therefore, you just have this dye to cyclic GMP, and look at the colors. This is 5-orange alone, and you add a cyclic GMP, you change its color, you can see this in the CD, the cyclic GMP alone, and then you then add your dye. You can see here that you are changing a lot of things. So this was our first contribution. It's a very, very simple idea. But the implication is interesting because now we have a fluorescent assay, so we can now go into high-frequency screening assays and find small molecule inhibitors. We also designed a second approach where this time we use a rival switch, which binds to cyclic GMP. And then we fuse this rival switch to another unit that bound to another fluorescent dye. And so we published this a year later. This is very sensitive. It can detect cyclic dinucleotides in nanomolar concentrations. May Herman, who was then at Berkeley, but she then moved to Utah, a year later utilized the same approach to detect cyclic AMP and all the different cyclic dinucleotides. And there is now a company. There is now a company that sells such rival switches that workers can buy these rival switches to detect cyclic AMP. So I feel that when we started, there was not a simple assay to detect these molecules, and we've been able to provide, very simple parameters to be able to detect these molecules. But I did mention that if small molecules can aggregate cyclic GMP, and if this is true, then one should be able to use the aggregation as a way to disrupt signaling. Cyclic GMP is important for bacterial biofilm formation. So therefore, if a small molecule can aggregate these molecules, then that means that would be a way to disrupt biofilms. Okay? So we made this prediction. We decided not to pursue this work because I became convinced that a planar small molecule that would bind to cyclic dinucleotides and aggregate might work in vitro, but it's going to be very difficult to translate that because they might also bind to DNA. So I was not convinced about the ultimate translation, but it's a key idea. So it turns out that this year, I found out that a group had actually taken this idea and run with it. So there is currently a paper in JMed Chem, was just recently published, where the authors said design and synthesis of a novel second PG quadruplex induces as bacterial biofilm inhibitors. And so the idea here is that when cyclic GMP concentration goes up, bacteria forms biofilm. And so if you use a small molecule to aggregate, then these aggregates are no longer biologically relevant, and so you can disperse biofilm, right? So this came from our aggregation paper, and they were very nice. They cite a lot of these original ideas, but here is the data that really excites me because the idea works, right? So here they have bacteria that has a lot of cyclic GMP, forms biofilms. They add a small molecule that forms the aggregate, and they can disperse biofilms. So sometimes in science, it's okay to come up with an idea for others to actually run away with it. But the whole idea that we could use a small molecule to aggregate cyclic GMP made us start to wonder, can we extend this to other dinucleotides, right? So cyclic AMP is also of interest, and it had already been shown by others, especially Nick Hart at Georgia Tech, that polyadenine DNA can also form a duplex, right? So when you have a lot of adenines, they can form a duplex at higher pH. But Nick Hart and others had identified this small molecule called coralline. That coralline was a small molecule, a dye, that would bind to a duplex that is only made of adenine, right? So it looks like the structure was just enough to fit into adenine base pair. So the question that we asked was, how do we leverage this for detection? Because unfortunately, this is fluorescent molecule, so it's highly fluorescent, but when it's bound to the DNA, it's also fluorescent, right? So it's very difficult to leverage that for detection. And so we came up with this scheme. So the idea is very simple. So here, we recognize that coralline and many other fluorophores are quenched with halogens. So like if you have a bromide or an iodide, it can quench your coralline, so fluorescent is quenched. But if adenine also forms complexes, if we can use coralline to form a complex with adenine, then maybe perhaps we would be able to separate the coralline from the quencher. And so here, you can see here that if you have coralline and you have a bromide or iodide, very low fluorescence, you add cyclic GMP because you form this inclusion complex, the fluorescent foci, right? So we've used CD to show this. So for example, this is cyclic AMP. You see the CD signature. You add coralline, CD signature changes, okay? But it is not just because it's got adenine. Look at PAPA is a linear form of cyclic AMP. When you have PAPA, this is the CD. When you add coralline to PAPA, this is the CD. So what I want to convey here is that when you have a dinucleotide, it appears that that cyclization endows the molecule with aggregative behaviors, right? And one can actually take advantage of this and come up with sensing paradigms. And so we did a job plot to really show that these are really interesting aggregates. So where do we take it from here? So the second major contribution was that others had shown using genetic methods that if you reduce cyclic GMP bacteria, it will lead to biofilm dispersal, and increasing what they call virulence and swarming. Similarly, genetic methods had shown that reducing cyclic AMP in gram-positives will lead to growth inhibition. But there was no small molecule. There was no molecule to use as a pharmacological agent. But we spent time coming up with all these detection methods. And so now we could go and do high-throughput screening, right? So we didn't have the tools to test if pharmacological inhibition would affect lifestyle. And so we used our method to demonstrate that pharmacological inhibition of these related proteins will field hopping, deletion, or overexpression. So this is going to be a new paradigm for antimicrobial agents, that we can now target these signalers to come out with new antimicrobial. Okay, so 2005, Howard established that low levels of cyclic GMP result in biofilm, high levels result in swarming. We now have a screen. So from high throughput, we screen several thousands of compounds. We identify this inhibitor as an inhibitor of a cyclic GMP PDE, and then we show that if you do a swarming assay and you add this small molecule, it actually inhibits swarming, which is almost identical to using genetic tools, right? So this was published in 2006. Likewise, cyclic AMP had also been shown to be important in gram-positive bacteria. It affects so many processes such as peptidoglycan homeostasis. Cyclic AMP has also been shown to regulate, to be an osmoregulator in gram-positive. And there was this really brilliant work by the Grande group to show that if you disable a cyclic AMP synthase, so this is this A, if you disable the synthase, show that bacteria become susceptible to antibiotics. And so what she showed here, which was a really great experiment, was that oxacillin, which is an antibiotic that targets the cell wall, the MIC to wild type Staph aureus is 4 micrograms per ml. She disabled the enzyme that makes cyclic AMP, and look at the MIC was down. So these are genetic ways, but of course, in the clinic, you need a small molecule. And we have a high throughput screen. And so therefore, the goal was that we could actually develop small molecules to inhibit the enzyme that makes cyclic AMP. And so we basically use our Caroline assay, right? We developed this assay, which I showed. We did a high throughput. We could find an inhibitor. So with that an inhibitor, there was no fluorescence. And so we showed for the first time that this molecule is an inhibitor of the cyclic AMP synthase. So here we have the enzymatic assay without inhibitor with an inhibitor. So for the first time, we have this agent. Again, would it work against bacteria? So this is the HPLC verification. You have cyclic AMP. You add a small molecule. You don't have cyclic AMP being formed. So this is GW5074. You see, there is no cyclic AMP. It's a true inhibitor of this enzyme. The question is that would it kill bacteria? Right? So antibacterial activity. Staph aureus, the compound inhibits Staph aureus growth. E. coli is a gram-positive, doesn't make cyclic AMP, the compound does not affect E. coli, right? Same with Listeria monocytogenes, the compound affects Pseudomonas, really does not affect the compound. And the MIC is about 2 to 4 milligrams. Okay, so the first part of this talk, what I showed was that cyclic dinucleotides are important in bacterial lifestyle. And when we started, there were no assays to detect them, and there were no small molecules to actually inhibit them. So we were able to provide that to the field. But it also turns out that immune cells, right, are always fighting bacteria. So now I'm going to move to immune cells, and I think that this is not a frontier of cyclic dinucleotide signal. So immune cells are insulted, right? They are being attacked by bacteria, DNA viruses, RNA viruses, and also even dead cells, right? Cancer cells, when they die, they are releasing DNA. So all of that is basically getting into the cell. And I'd like to mention that the reason why COVID is partially so dangerous is not necessarily because of the bacteria itself per se, but some patients succumb because of the inflammation after getting COVID. Okay, so inflammation is a big deal. So let's see what happens when foreign DNA gets into cells, into mammalian cells, right? That is a bad thing because DNA usually resides at 2 places, right? So DNA is either in the nucleus or it's in the mitochondria, right? So if DNA from the mitochondria gets into the cytosol, it means that there's probably some damage. So that's a bad thing, right? But if DNA also comes from the outside, if it's from bacteria or virus, it's also a bad thing. So it turns out that mammalian cells have evolved to be very good surveillance agents, right? They are always surveilling their cytosol for things that should not be there. So if DNA is in the mammalian cytosol, then there is an enzyme called CGAS. Okay? So CGAS will bind to the DNA, and once it's bound to DNA, CGAS is an enzyme. it will take GTP and ATP and make a cyclic dinucleotide called CGAM. This is the mammalian CGAM that I showed, right? So it's produced in response to a foreign DNA, which is usually a danger signal, right? Okay. Once this cGMP, which is the mammalian form, is produced, it binds to a hub protein called STING. Okay, so STING then binds to CGAM, it gets activated, it's trafficked to the ER, where it binds to this kinase called tan-binding kinase. That then phosphorylates IRF3, which is a transcription factor. IRF3, once it's phosphorylated, dimerizes, and then it goes to the nucleus to produce IFNs, right? Cytokines. So basically, this is the way that a cell senses the danger. It produces these inflammatory cytokines. These are then pushed out, and then they activate the immune system, right? So cytotoxic T cells will come to the area and take care of the problem. But bacterial cyclic GMP and cyclic AMP and also CGAM from bacteria with a 3', can also bind to STING, right? So this hub protein can detect the mammalian cyclic dinucleotide, the bacterial cyclic nucleotide, and basically mount an immune response, okay? So this is currently the picture. This is what we now know about STING signal, okay? So we've always wondered that if mammalian immune cells are this proficient at sensing pathogens, then how come some pathogens are able to actually go inside the cell? So for example, TB, right, tuberculosis, it's a highly successful pathogen. It actually gets into macrophages. How does TB go into macrophage and survive in that macrophage? So what that means is that there should be some strategies that the TB bacteria has evolved, right? One can predict that maybe the TB bacterium has evolved strategies to disable the CGAM STING pathway, right? So that was one of the questions that we asked. How come some pathogens are so successful when all of these actually take place? So our third major contribution was to elucidate how some intracellular pathogens are able to invade the STING program. So in collaboration with William Bishire, we published a paper a few years ago that showed that if you take TB wild type and you basically infect mouse, so this is a wild type, right? By 33 days, all of them are gone, okay? TB has an enzyme that we predicted to be a phosphodiesterase that will degrade the mammalian CGAM. So mammalian CGAM is the second messenger, right? Once it senses pathogen DNA, it makes this molecule to turn on STING. So we just had a hypothesis. We said that phosphodiesterases in the bacteria that would be able to degrade the host would be a virulence factor. So we basically disable that phosphodiesterase, right? And then look at that. This is wild type, and this is the bacteria without that phosphodiesterase. And then we complemented it back. And then we almost got back to wild type. Okay. So this was the evidence that the cyclic dinucleotide is playing an important role, and the pathogen has evolved to find an enzyme. Well, it's co-opting because it also uses that enzyme to break down its own cyclic GMP. It's co-opting this enzyme to basically. So we came up with this model. The model is that intracellular pathogens invade, they are in the phagosome, their DNA escape, they bind to C gas to produce cGMP. Remember, the C-diAMP of the intracellular pathogen also comes out. So these are all danger signals, right? The danger signals will activate STING. STING will then turn on interference, which is bad for the pathogen. And so what then happens is that the pathogen basically has an enzyme. as the host is trying to mount the challenge, the pathogen is also destroyed in that challenge. So if you are a drug developer, you take all this information and then you say that, okay, that means that this is a target for antivirals. So let's go and find small molecules, right? Again, we develop the assay. We can now find an inhibitor for almost any cyclic dinucleotide enzyme. We've spent the years to build that. So we went back and we said, okay, let's develop an assay. So this year, we basically reported that these molecules inhibit the TB enzyme. So these are going to be antivirals. So what next? So after we put out all of these things out there, we started to collaborate. So I'm collaborating with Franbian at Indiana School of Medicine. He works on Borrelia that causes Lyme disease. It turns out that both cyclic AMP and cyclic GMP, very, very essential for tick colonization and also malaria infection. So we hypothesized that small molecule inhibitors targeting cyclic GMP would eliminate Borrelia from ticks and malaria. This called a 4%. The reviewers loved it, and the reason is that we spend all these years coming up with assays to do that, and it's also showing that once one hits these enzymes, you can actually disable bacteria. And I'm also pleased to mention that we have already found inhibitors for these enzymes. So Fran is now doing the bacterial work and very soon for this. Okay, so it turns out that sting is this interesting how protein is found in autophagy. That will be another talk. It's found that sting and the IFN helps in immunity towards cancer. Okay. So our fourth contribution, and I think perhaps this is the most important contribution. So collaborating with the Graham's group at Albert Einstein, we were actually the first group to show that when you activate sting, you can actually cause cancer immunotherapy. This was a very difficult paper to publish. But once it got published, there are now many companies that are now targeting sting. There are many sting agonists in clinical trials. Okay? And I'm just not saying this, I have the publication to prove it. This is our paper. We published it in 2014. Okay? So we treated mice with metastatic breast cancer, and then we immunized it with cyclic GMP. And we were able to show that this actually immunizes the mice and the ability to grow. And so this is the basis. It's possible that other people were working on it as well, but at least this really demonstrated that these cyclic dinucleotides are really important. Okay, so we developed cyclic GMP, cyclic AMP. It's something we bothered with because we could not detect the human form because the human form doesn't aggregate, right? But the human form binds to sting. So it was like the light moment came, the light bulb moment came, and we thought, gee, if all cyclic dinucleotides bind to sting, then why not just use a fluorescently labeled cyclic dinucleotide, mix it with sting, right? You get fluorescent polarization. And then you displace that with any cyclic dinucleotide. This is a general assay for all cyclic dinucleotides. But what this meant was that now it means that we can find an inhibitor for sting, we can find an inhibitor for all the phosphodiesterase that create the host cyclic dinucleotide. And so cyclic GMP is a data signal. EMP1 is a target, an anti-cancer target. We are developing inhibitors for EMP1. Sting, although activating sting is good for cancer immunotherapy, right? Because it leads to inflammation. What it also means is that if sting is activated, it's also a bad thing. So there are some patients where they have activated sting and they have very, very serious inflammatory diseases, right? Okay. So this is one example. So what that means is that there are also people looking for sting antagonists, and It has also been shown that inhibiting STING actually mitigates acute kidney injury. I'm showing all of this to sort of show that this STING protein is really important, okay? And so currently, another ongoing research, I'm just giving you a flavor because we have all these platforms. We just discovered a compound, HSN1077. It's basically inhibits STING activation. So this is cyclic GMP. When you add it to macrophages, you see the IFN expression. But when you add our compound HSG1077, you can actually plant STING, right? So that is something that we think could be translated into immunotherapies. And I don't have time to go through this, but we are also looking at nonstick pathways, okay? Because I think that nucleotides are so important, we believe that they go through nonstick pathways. So we use proteomics to ask the question, are there other receptors apart from STING that also bind to cyclic nucleotides? And so using different cyclic nucleotides, I'll just rush through this. We did a proteomic study. I'm going to rush through this to show that we just reviewed that cyclic GMP, the bacterial cyclic nucleotide, that we had earlier in 2014 shown that we could immunize mice. Actually, upregulates HOX2. HOX2 is not a good. It's an inflammatory molecule. We don't want too much HOX2, okay? And we have been able to now provide a mechanistic. So we did some mechanistic where we show that just cyclic GMP goes through the leak-egg pathway to regulate CREB, that then leads to HOX2 upregulation. Okay, so this was just published in ACS. All right, so I'll just finish by saying that our major contributions to this area have been the pharmacological inhibition. We were the first to show STING activation as cancer vaccination. We've shown host-pathogen interactions, and what we are now really focused on is these non-STING pathways. Okay? For this talk, I only talked about our cyclic dinucleotide signaling, but we also work on kinase inhibitors, G5 antibiotics, etc. We collaborate with many different groups. We have a company now that is hoping to translate very potent leukemia inhibitor. So gilteritinib is now one of the best-in-class drugs for acute myeloid leukemia, but there is resistance to gilteritinib. So we've shown in a gilteritinib resistance, this is our compound, KinaRx compound, which is doing so well that we hope to take this to the phase one trial soon. We work on quorum sensing bacteria talking to each other. I didn't have the time to talk about it, but we look at biofilm maturations. And in collaboration with others, we saw the crystal structure of transcription factors that actually respond to quorum sensing, and other proteins as well involved in quorum sensing. So this is a picture of my group. Because of COVID, we don't have a recent picture where we are all in the same group. This was the last time that we had a dinner together, and little did we know that it will take 2 years for us to come together. All this will not be possible without generous funding from all the funding agencies. So at the beginning of my career, I'm very, very grateful to University of Maryland and the Camille Dreyfus and American Heart Association. But recently, our work has really been funded by Fluorescent as a national institute. Okay, so I'm happy to take any questions.
Any questions?
Hi, very nice talk. Thank you for that. So I have a question about phosphodiesterases. I think at the center of everything that you show is phosphodiesterases for different cyclic nucleotides. You are showing that there are different PDEs. So I'm just curious to see, how similar these PDEs are. For example, you said that Mycobacterium tuberculosis PDE can break down the host cyclic nucleotide. So is there studies on that, and are people trying to engineer PDEs to see if they can, make break down different cyclic molecules?
Yeah, no, it's an excellent question. So we started this study, right? So what we are noticing is that the PDEs of the intracellular pathogens tend to be more promiscuous. And there might be an evolutionary reason for that, right? Because they also have to cleave their own with the cyclic dinucleotide. But the PDEs of the other bacteria are not. So for example, in Pseudomonas, there are actually 27 different PDEs for cyclic GMP alone. And we found small molecules that would inhibit one or two, but not the average. So it even shows that even within one species, the active sites are different. But there will be some PDEs that are more promiscuous. I don't think I've seen any paper where anyone has actually done more systematic study. But it's just that we've seen this correlation that intracellular pathogens tend to be more promiscuous. Now, with your other question, which is a brilliant one, is if one could engineer that, right? So people are engineering a cyclic GMP synthase and then using that for cancer therapy, right? So bladder cancer, for example, one of my collaborators is currently working on that for bladder cancer. But not the PDE. The PDE could be interesting for, actually I take it back. I was gonna say that it could be interesting for the inflammatory stain related diseases, but some of them are constitutively active even without a cyclic dinucleotide bound. So one actually needs a real stain inhibitor. Right, yeah.
Can I just ask small other thing? And for your high throughput screens, the libraries that you use, how big are those libraries and what is your success rate typically?
Yeah.
I know that.
The biggest screen we've ever done was 200,000. And out of the 200,000, we probably got about 50 hits. And then when we did the HPLC triage, only about 5 or 10 were unique structures. And so one of the things that got me really disappointed was the low quality of the chemical libraries. So we get so many hits, and then you look at the structures, and some of them are like PAINs, pan-acid interference agents that I don't even know why they are in the library. And then the majority of them are close to the rate. So 200,000, we probably will have about 5 unique structures, and then we then go ahead. Yeah. So it's not just the number of the compounds. I think the quality of the library is more important.
So evolutionarily, are the bacterial cyclic GMP cyclases related to C-gas? Did they have similar structures? Did one evolve? Did the eukaryotic evolve from the bacterial?
Yeah, no, it's an excellent question. There is some, yes, excellent question. There is some similarities, but also different. Because even when you look at the bacterial synthases, there is a lot of divergence, right? But there are some C-gas-like proteins that have also been found. In bacteria too. And it's very interesting, not all the C-gas-like proteins actually are enzymatically active. And it's not always that because their active sites have been mutated, right? And so yes, there was a recent Nature paper that came out that is looking at linkage between C-gas in mammalians, in fungi, and also trying to compare that to bacteria. But that is really at the end stage. But that's absolutely. Thank you.
Yeah, very interesting talk. So you showed us in these last slides that STING is essential for these regulatory pathways, but in therapeutic point of view, it is a kind of two-sided sword, because you could eventually boost it and fight cancer, but you might induce this autoimmune diseases at the same time. So therefore, ideally would be a drug which only kills the cancer cells without having this negative effect of the immune system. Do you think that such a drug can be developed? And also my question is now from the structural point of view, what's known actually about the structure of STING? Is there a crystal structure of it or a cryo-EM picture? How big is it?
Yes, yes. So I'll answer the first and the second. So excellent two questions. Initially, right, when our paper was published and then a year later, Aduro Bioscience. So what Aduro did was that they took cyclic dinucleotide and they made a phosphothiazide, which is more stable to nucleases. And that's what it took to clinical trials. Aduro was actually bought by Novartis. Novartis gave Aduro, I think, 100 million upfront and up to 750, depending on milestones. The clinical trials it didn't really work, right? And you've just hit on the point that if you activate STING too much, you might have that inflammatory issue, which for certain late-stage cancer patients, that could be seen as a side effect, right? And so that was not maybe so much of a concern, but it's actually turning out that there is even a bigger problem. It's turning out that STING is this double-edged sword where at the beginning, it's okay, but if you activate STING for too long, and if the environment becomes too inflamed, then what then happens is that you actually help to degrade the matrix around the tumor, and you actually promote metastasis. So it's now coming out that STING is this dual thing that, right? And so the jury's out there that maybe perhaps STING inhibition as a cancer immunotherapy is something that is more nuanced, and we need to be aware of it, right? So that's the first. Your second question is the crystal structure. So yeah, the crystal structure of STING has been solved. It's got a transmembrane pad, and then the outside is basically, so STING is a dimer. So it's a dimer molecule. It almost forms something like this in the inactive form, and then when cyclic dinucleotides bind, it basically closes. So that has been solved.
But not with a membrane anchor.
So
But not as a membrane-bound form, correct?
they used the membrane one was also done. I think they used it wasn't X-ray. It's escaping me. Cryo-EM.
Okay, for the
There was a
full length
cryo-EM.
structure, yeah. So do they also solve it in combination with drugs already?
So
So do they also solve drug complexes with STING?
Structures with the drugs.
with the
bound?
drug, yes. So they've solved, yes. So structure of STING with cyclic dinucleotide bound has been solved. Sorry, I misspoke. Yeah, structure of STING with cyclic dinucleotide bound has been solved as a full structure. With the small molecule inhibitors, they've solved it with X-ray, not with the membrane pad. Just the extra, yes.
Thank you.
Do the toll-like receptors get induced by these like the nucleotides from these bugs?
So they don't.
No,
So they
it's
don't.
not part of the
They
natural.
don't. Yes. So it's not like CPG, right? And LP is not.
Okay, I was wondering if that was. Any other questions? Okay, well, let's thank Dr. Sintim one more time.